to be here with you all. This is the second to last webinar for this season of the International Legume Society webinars. We are here today with Dr. Dil Thavarajah, that comes from Clemson University. She's professor of food quality and nutrition breeding, and today we are going to talk about global food security. So, Dil, please.
Thank you. Let me get share. Can you see my slides? Okay, perfect. All right, thank you everyone. And today I'm going to talk about pulse crops towards global food security. But really, I don't have much of the work about the global in here. I've done a fellowship in Washington, D.C., USAID for Middle East. But I'm not showing that data, but I'm going to show most of the work that I am doing for my program, which is a pretty new breeding program which started in 2020. And it's 5 years. And we are in a progress with 100% USDA-certified organic material. So I'm going to talk about what we do here at the Clemson University. And get that page down. Okay. But actually, I want to go up because Dr. Wackington is in there. I wanted to show, and this is one of the crosses that we made that he gave us the best variety performing in South Carolina, the CDC Inca. So we made a cross. So I put that picture there because to honor him and giving us the material to start our program. So some of the Canadian material works really well in South Carolina. So I will talk about a little bit about it. So before start anything, I like to put the disclosure. I have no financial interest or no conflict of interest, and most our funding is from USAID. It used to be, and then now the federal government, National Institute of Food and Agriculture. So I have no financial interest. About the outline, I'm going to talk a little bit about agriculture revolution, and then we will move into the food system approach. That's what my program is about. And then what biofortification target, because I was trained as a biofortification, more on the nutritional traits and the physiology. So if somebody asks me about pathology, don't ask me, I don't really know. I work with the pathologist. And then we'll talk about the Clemson University Pulse Breeding Program, what we do, what barriers we have to adopt the organic, because my program is 100% USDA certified organic. And then our partnerships and collaboration, what is our toolbox look like, and then our impact assessment. So let's think about the agricultural revolution. Where are we at in 2025 right now? 80% of our diet, if you look at our diet, it's about 12 dozen of crops, mostly wheat to barley. We all like beer, and then legumes, and also the oil crops like fat and flax and canola. But actually, canola is not part of the Neolithic founder crops. But if you look at the peas, lentils, and chickpeas in there. So peas are my most favorite crop, and it's very easy to adapt for South Carolina. Lentils are chickpeas are on the way, the pipeline, because of the humidity and the high heat. We have becoming a very hard timing, get the successful crosses compared to the peas. So with that, let's think about a little bit about the Green Revolution. The first one is Robert Bakewell. So who is the agronomist in 1700, talked about breeding and the animal breeding and the agronomy. Moving to the 1866, our father of the genetics, the Greg Mendel. So moving from 1700 to 1866 to 1970, the father of the Green Revolution, Dr. Norman Borlaug. Our students always go every year in the November to listen about the Borlaug talks in Iowa. And we are emphasizing this because I used to teach the global food security class here at Clemson University. So we emphasizing what he has done, how much his research impact on the reducing the global hunger. So with that, what do we see today in 2025? So you can see the first graph, the population increasing very rapidly. But then when you think about your food, this is about 61 to 2009. You can see per capita pulse production, that is peas, lentils, and chickpeas, kilograms per capita per day, it's reducing. Actually, in US, it's like people eat minimum like 1 cup. It's not a very popular crop in South Carolina. It's very hard to get this crop or get this food into the plate for the new people to try it. I'm originally from Sri Lanka, so it's not a big problem for me. But you can see how the crop supply, but the fat and sugar, we all like our donuts, we all like our pastries. So production of the sugar going high and the pulse production is going down. And this paper coming from one of the science publication, you can see there is in 2050, there is a big yield gap with this way that if we are how we producing with the looking at the population increase and the most nutritious crop supply go down and most wheat and other crops we need energy production go high, so there is a yield gap. So if there's a yield gap, we might have to face for the consequences in many things. One thing is we can't feed the growing population. Also, what would be the nutritional consequences? So looking at the UNICEF, this is the recent data from 2021. You can see that 149 million children age under 5 are stunting and wasting and overweight. So this is stunting, wasting, overweight related to micronutrient malnutrition. But what about the obesity and overweight? So you can't really say, okay, you are very healthy, but you're overweight or obese. So that also come under the malnutrition. So 2.5 billion adults are now obese or underweight. So why the reason? Because is something changed, right? Going back to my previous slide, our food consumption pattern has changed over time. And here is another consequence. So these are pictures from micronutrient deficiency, calcium deficiency, vitamin A deficiency, rickets. One of my graduate students now work in Boston Children's Hospital as a pediatrician. He's working on how the diet can be a part of the preventing rickets and the vitamin A, and they are promoting a lots of pulses to provide these kind of nutrients. So can we, from 1970s to 2025, most of breeders are here, pathologists and scientists working on pulses. Can we think about a little bit more on the revolution to go to produce safe, sustainable, nutritional food for all of us? So that's kind of where we're going at right now. And for that, I designed my program with the old literature from my mentor, from Dr. Combs, shared a lot of advising for my program. So we are bringing their old concept from 1996 into 2025. What we are doing here is actually production or plant breeding is it one section of a whole food system. So how do we build our food system around the crop production? So crop production, acquisition. So everybody need to have a safe, nutritious food. And also they need to know how they can utilize it, how they can cook it, how they can use it, what flavors it. So with all of that, we design a program which is nutritional breeding, more emphasis on the nutritional traits, which are very hard traits because these are quantitative traits. They are controlled by more than one gene. Most of the breeders, they like to concentrate on not very many, like the most varieties that release from Canada has very high protein content, bioavailability, that's good. But then we think about more of these are very hard compared to the yield because our focus earlier was more for the yield, feed the people. So now we wanted to have more protein-based micronutrients. So Canada has done quite a bit of work on this part of the biofortification process. I'm not going to go talk much about pulses because this is the legume workshop, so everybody knows about legume. But I want to emphasize one thing about we call pulses as the poor man meat because I go to the grocery store, I can buy a pound of lentils for $1 or less. And but if I go buy organic, it's actually $2. So it's double the price. And that's also like a very important point for our growers. If you grow organic, you will get double the price. So organic chickpeas, lentils, and organic peas are very expensive because they are, for the amount that they get, it's expensive compared to the conventional. But pulses are really beautiful crop because it's not only giving the 100% human nutrition benefit, but also it gives the ecological as well as the environment benefits. So that's why we thought this is exactly fit for the organic agriculture, not only for the yield, also we can provide vegetarian people 100% organic plant-based protein, which has about 80 to 85% digestibility. So when you look at the seed of lentils, it has 60% of carbohydrate. But when people say carbohydrate, they're scared. But don't be scared to eat pulse carbohydrate, which has about 20 grams out of 60%. 20% is prebiotic carbohydrate, which release the sugar very slowly. Even starch has different properties compared to the others. And the protein is absolutely wonderful, 25 some of the varieties released from Canada has about 25% of protein in peas. It's very high amount, so you need 100 grams to get 25% of dry weight. So when you cook, it's a different story, but still it's better than, if you are a vegetarian, it's a better protein than eating the red meat. So let's talk about my state. I'm from South Carolina. I work here at the Clemson University. So this little state down here, but this is the whole map of the United States. So you can see all these squares with the purple. That is the Midwest. Pulses are home for Midwest. That is North, South Dakota, Montana, Palouse, California regions, and parts of Iowa. But when it's come to the East Coast, you can see that these purple little blobs. Those are actually not peas or lentils or chickpeas. These are actually they are beans. It's a little bit of dry bean, and also they make cowpeas. But so our goal is, can we have a winter crop from East Coast, from New York to Florida? Our winters are not snowy like your winters. Our winters are very mild. It's like the Mediterranean weather. So our temperatures range from 15 centigrades in January to May is like 80 Fahrenheit. That means like 30 centigrades. So we have like a very nice winter that we enjoy a lot. So here is the map of South Carolina. You can see the commodity map. It is all about soybean, peanuts, and the other commodities like eggs and chicken and all that, corn, soybean, and peanuts are our major crop. But we are here for about 25,000 farms, and it's a very green place. But the problem in the winter, they don't have much of the very many options. Winter means from January, right after Thanksgiving, last week of November, December to May, what they do is some places they grow winter vegetables, but most of the places they keep the land as fallow, do not grow anything except keep it as a fallow, or you put the grass or a legume cover crop. That's all we do. So what we thought, let's have a winter cash crop so then growers can have a amount of money selling these peas, because most of these peas goes to the noodle factories and also dog food and also plant-based protein. But there's a high demand, but there is not enough production here in South Carolina because farmers don't know, and this is a very specialty crop. We call it as a specialty crop. But then the conventional breeding as well as the USDA-certified organic, I have a conversation with Nelson and Dr. Asid about what is USDA-certified organic mean. So USDA certification takes 5 years, 3 years to the transition, and then move into the certification. So there's 5 things that we need to follow. Is we have to follow the federal standards. So the federal agency agents will come and audit our programs and then make sure we are not using any GMOs, we are not using any synthetic fertilizer, and we do not do any radiation or mutagenesis in our program. So these 5 things had to be go through it, and then they record it, and then that's how the whole process going on. So once the land is organically certified, if you add any fertilizer without having documentation, USDA certification, your certificate will be gone. So farmers are very careful, and these are not large farmers like Canada or the Midwest of the US. These are small scale farmers. They have acres range from 100 to 500, a few large growers, about 2,000 acres, but they are only very few. Most of our organic growers are smallholder growers. Some have 2-3 acres up to 100 acres. So, but we strictly follow these 5 guidelines. And my program at Clemson University, we do many things. The breeding program is very, very new. It started in 2020. I work with the breeders around the world, as well as we exchange the materials. And the material that we develop here at the Clemson University will be tested in Spain as well as in India, because we wanted to make this as a collaborative project. And so then we can do organic research part of the with several breeders. And mostly that our emphasis is for the nutritional traits, and we work with the pathologist and helping us with the disease identification and things like that. Work with the ICARDA and also Spain. And most important part of our program is not only this variety development, we looking into the consumer profiling. So we do taste testings and the sensories at early with the production material from the market, and then we ask the perception of the consumers, and then we design our program based on their perception. And then we, at the end, we again do the sensory, we do cooking, and we bring the chefs, and we do try to bring that into our varieties, more add the flavor into our crops. And then extension is a very big program of my program, because I have a 3-way split duties for my job. So we do going organic platform. So we educate farmers, we educate high school students, undergraduates, and new crops. These are mandates from the land grant mission of our university. So we do quite a bit of extension activities. Also, we teach healthy home gardening for the people who don't know about healthy diets. So we try to learn, teach. We learn from them their traditional activities of how they eat. Also, we teach them how we can adapt the new things into their dietary plate. So here is our breeding pipeline. So we have trade discovery, and we have a team of people. I work with the Cornell University and the K-State University, also Texas A&M. And we have a team of people help us with the trade discovery, population improvement, and we do the product development, which is like, I really like that part. And we do integration. Also, we do the participatory breeding with the farmers. Everything we do, we don't do it in the research fields in the Clemson, only for the testing. We go to actual farmer, we use their planters, we use their, I have a planter and a combine and everything, but we take them to the, do our advanced trials. But mostly we try to use the farmers' equipment, parents and teach them how to do that. So this way they can use the crop. And we start with a large amount of material. And then within 5 years, we are right now in F9 stage, and we are writing to release our 100% organic varieties with the on-farm trials. So this is how the breeding program looked like. So this is a picture of the germplasm that we collected from parts of the world. And we tested, look at our soil. And this is organic land, 100% organic. We did not spray anything. It's all about the management. And then we select our parents, we make crosses, and we do single seed descent method, and then we evaluate the varieties. But before we actually made our crosses, we do the sensory panels and the consumer panels, and then we make our food products in the same program. So it's from a more food science component, nutrition component included, embedded into the plant science program. So we have a couple of bioinformaticians help us with the genomic platform, and also we have a phenomic toolbox for the breeders to use from pheno apps as well as to the drones to the nutritional traits. And then we looking at the nutritional traits like milling, color, flavor, texture, aroma. As breeders, I know when you have a large amount of samples, it's very difficult to measure these traits. But now we develop tools that we could measure 5,000 material decrease into like, I don't know, 100 lines by doing our toolbox. And then the end quality is very important for us for the protein digestibility, amino acid balance, and also the nutritional value. So these are the targets that my breeding program that we set for the breeding, and we cannot do a lot of yield compared to like the conventional, but we are pretty happy. Our yield are about 1,200 to 1,500 kilograms per hectare. Like it's because of the organic management and the fertilizer. But some of the lines that we made with the Canadian Dr. Walkington's program, they yield up to 1,800 kilograms per hectare, but it's hard to get 2,500 kilograms per hectare like the conventional. But it's okay. The yield is about 1,500 to 2,000 because the farmers get double the price compared to the conventional. And then we do all of this. We wanted to have a 90-day crop, and we are successful on that. Plant height, all of these traits that we are working on it. Here is look at our organic fields look like. So before we planting, they have a cover crop. So, and then they're crimping when they are 60 days old or 50 to 60 days old. And then once we crimp it, we run through the plow and then level the land, because otherwise the weeds can come up. So we make sure the weed emergence not happen. It's take 5 years to reduce the weed emergency and or the germination capacity of the weed seeds. So this is our new GPS a planter that we have, and that is Tristan Lawrence. He's planting, and we used to have a very old planter from the cereal breeder. That's what I used in the very first time, but now we have everything GPS, and we do not add anything. It's just the land preparation, and then we plant. We have a microwave heating weed controller. It's like the AI control one. We did not use in our program, but so that's also possible for the weed control. Okay, page down. Okay, and I wanted to talk about the our toolbox for the phenomics toolbox. So this is Tristan Lawrence, my project manager, and this is Amod. He's a senior PhD student. Amod and Tristan, basically Amod developed the whole phenomic platform for his PhD thesis. So what do we do digital monitoring here? So I used to go with my pen and paper. So now we are really advanced. So we have the Pheno app with our tablet, and also we have drones that fly on 20 minutes that can get our data, but we still like to walk in the field. So what we do, we measure the plant height, days to flower, canopy closure. Also, we bring the seeds to the lab, and then we measure the nutritional traits like carbohydrate, proteins, fat, and the flavors. So what we want is to develop like a precision agriculture to reduce our time in the field. This really helpful because you drive 2 hours, 2.5 hours to the field, and you drive back 2.5 hours. It's like 5 hours on the road. And having this is very useful because 8 hours you work in the field, come back very tired. So this toolbox is so I recommend for anybody who wanted to start a new breeding program, just use these tools. So this is the new, so in our country that we have to use the US manufactured drones. So this is Skydio X10, very cool, cute, small drone. So this is Amod running through the drone in 20 minutes flight time in my breeding lines. These are F8 trials at the cherry farm, one of the organic farms in Clemson University. So this is our a new one. Looks like it has a 50 MP camera and also RTK module. So it's actually automatically capture the high resolution 2D model in our controller. So it's like, and then I'm going to download the control into the cloud system so then I can watch that from my computer. It's so cool that saves lots of our time. And it's not that expensive. I think we one of the grants paid for it. It was 21,000 US dollars. So how the modeling look like? So these models were developed by Amod for his PhD thesis. So he get the 2D maps, and then he stitch it with the platform developed by the Texas University Dr. Seth Murray's program, and we collaborate with Dr. Seth's program, and Seth taught us, and then we wrote the coding. Amod wrote the coding, and then we can actually very precisely do this for canopy height as well as the plant maturity, days to mature. And also we are helping this system for our growers. So these are kale plants. So the largest kale grower in the United States is actually in South Carolina, WP Roll Farm, where I do my work. So so what we do is we're helping with our phenomic toolbox that they can, from their computer, they can say what time they can harvest their kale. So, and then we train the growers to run their drone and upload the models or upload the pictures into our cloud system. So then we can analyze their data and they can have it for their farms. So it's as a state program that we help out our growers to have a better crop. So this is my F8 from this year. Amod actually stitched this last night because he's working all day on the preparing because he's graduating in December. So this is very interesting. We have been working on this for last 3 months, and then this is from the germination stage and then vegetative stage and the mature stage, as well as the fully mature stage. So all these individual data points, I have it. This is like the big picture of that, but I want to show you how successful that platform. And then that is in the field. So what happened in the lab? I have a very nice phenotyping lab, which has everything, all the instruments that I dream for in my career. So I have everything, but all these analytical instruments are very expensive. Running a sample for the protein is like for amino acid, it costs us about $100 to $200. Run a nitrogen, it cost us $10 for the soil lab to run through the LECO. And the digestibility requires enzyme kits, and which take so much of time. So with part of the Amod's PhD thesis, we developed technology called combination with the Agilent Technologies. We partnership with them, and we create the FTMIR, Fourier Transform Mid-IR. So you can see this is Sonia, one of my senior PhD candidate. He's she's having a 1 lentil seed, and then we measure here, we can get 8 traits from this bench top model. And then now right here, Amod is holding the 4300 carry handheld FDMIR unit. We can take this to the field. When the plants are ready to harvest, we can split open a pod and we can get our nutritional traits. So he was very happy that he could incorporate that into his PhD program. So the day that we got this from Agilent, that was the best day for our breeding program. So here is the high throughput phenotyping platform that we use, the FTMIR. This unit is about $25,000, I think, from Agilent. So in 2 minutes, we get all these traits, but now actually it expanded to other PhD students also working on different traits. So one student only working on flavor and the other student working on the physiology part of the leaves and the roots. So we are trying to use this technology to do our monitoring of our nutritional traits. And this is a picture of the diffuse reflectant mid-IR. So this is the handheld unit that we got from Agilent. And it also, same as the benchtop, we can measure all these traits in 2 minutes. But the beautiful thing is it's heavy though, but we can take it out to the field and we can mark which one to harvest and which one not to harvest. So this is like how it works, that I want to show that almost it's holding 1 seed, or you could do like a whole bunch of seed. You could do this in the feedstock. You could do in fruit juice, anything like that. So he's developing all the models. And this is how we work with our growers and our partners. So whoever has these instruments, they can measure their traits and they can upload into our cloud, and within next couple of minutes they will get exact data how they want. Or you can collaborate with me. You have sent me the seeds, but if you wanted to do it in your lab, this cloud system has the models, so it's automatically generate the numbers, and then our phenotyping group will create the data for you. So moving towards the genomic tools, so we have 2 people, actually 3 people in our program. Dr. Broadwright, he moved to Corteva, but he developed the platform for us. We create the large set of germplasm for all 3 crops, and then some of the material for the genotyping that we used, the public material, and also we did the whole genome sequence material, which is available from our breed base for all the breeders around the world. So we are 3 PhD students on this project. So this is the protein diversity panel that we got from ICARDA to measure the protein quality, amino acid quality, and the digestibility. And this is a little bit of data from Sonia. She has not published that, but she assembled a lentil diversity panel with the 450 individuals, and some data of the genomic data available from the global platform, from GRIN as well as from ICARDA, but rest of the things that we genotyped, and it's going to be available for all the breeders around the world. We are going to put that in the USDA database, so then everybody can access to this genomic data. And then this is the new crop that we are going to adapt to the East Coast. And I'm working with Dr. Dr. Vandermark. He's the chickpea breeder from USDA-ARS. Mostly chickpeas here we grow as daisies. Kabulis is not very well adapted to our weather. So we are cleaning these days. We are planning to purify billy beans because billy bean is well adapted to South Carolina. Also, we are having a couple of lines ready to go with the daisies. And this work is also published about the chickpeas. We have a mini co-collection, and then it was genotyped by Vandermark group. And then our bioinformatics group ran on the minerals and the protein, and there's more work to be coming. And here is the IP management practice. So when we have these lines, these are organic. So we want to make sure these proteins are good, or these proteins actually has a what kind of properties. So we have a utility patent for our extraction of the organics. Remember that 5 rules that we have to use that. So and our protein is 100% organic and allergen free, no off flavors, and it can be application for the semi-solid form or baking or milk. So this is the aqua form that we get from the peas, and this is actually pea milk, and this is the yogurt formations that we did out of the peas, and this is the chickpea protein that we isolated using that isolation application pattern. And also this is some of the work done with the carbohydrate ingredients. We also separated the prebiotic carbohydrate as organic with the organic processing utility pattern. So this also part of our program. And then this is something that I started for last 11 years. I've been in Clemson for 11 years. So this is like the educating our community to eat healthy, a small place, a better place. So we call it Tiger Garden. We are Clemson Tigers, and we are famous for the football. And then anything with the tiger, it runs. So if it say the Tiger Garden, people love it. So actually, this concept started when I was a graduate student at the University of Saskatchewan, and this is one of the lovely professor that I go and talk to. He has this rooftop garden in the building that where I did my education. So he grow these tomato plants. Saskatchewan has a lot of winter, so if we can grow in the summertime this, that is a good concept. So I was thinking like this should be something that we should teach our young generations. So, and this woman from Ghana that convert her old well into a garden. You can see that how balanced it is. It has amaranth, it has cabbages, it has corn, bananas. So it's like any small place that you can convert into something beautiful. So here is the one of the school. It's Pendleton Elementary School, which is not very big school, but these kids, they don't know what vegetables seems to be, like how healthy they are, how do you eat. So my undergraduate students, they went and started a small garden there. So this is the community helping them to build this garden. So this beautiful garden, they built in the school as well as they built behind my lab. This is my lab, you can see in the back, and these are the garden behind my lab. So we use this as our model garden. Anybody who come and see, then this is beautiful kale. That's my pet project, pet crop that I really like, kale. So we grow different types of kale here. And also we do some breeding aspect about kale, which is totally different from peas in the greenhouse. And in the summer, we have a truckload of sweet potatoes in our garden. So what my take-home message is you can grow anything in these little beds. So we grow peas, lentils, chickpeas, sweet potatoes, herbs, and because South Carolina weather is perfect year-round, we can have vegetables. So our platform, you can see we working on 3 other crops. Also, our major idea is to increase the nutritional traits, and we bring this phenomic toolbox. And many people in my group, they're working very hard to bring this into the community level. And we wanted to make pulses better for American consumers as well as the global consumers, especially with the sensory, flavor, and cooking. So these are my team, and wonderful people, lots of PhD students and people help with the breeding. I want to emphasize Dr. Emerson Scheib. He was the soybean breeder from Clemson University, retired and came to my program, which is a wonderful asset. And then we have partnerships with the many parts of the world, and especially the large growers in South Carolina. Also, program has an advisory board. And also I like to acknowledge the USDA National Institute of Food and Agriculture. They support our breeding program, funding us with $1 million, and South Carolina Department of Ag. Global Food Shot and USAID. Thank you so much, and that's all I have. I'm going to
Thank you,
stop sharing.
Dil. Thank you, Dil. It was a very nice presentation full of interesting ideas. Now, if are there any questions from the nice people connected? Okay, we have Dr. Mohammed Azimazad. You are muted. You are muted. We cannot hear you. Dr. Azad, your microphone is off.
Okay. Sorry. No, it's okay. No, it's okay. Thank you.
Sure.
Because you are working for organic chickpea, so organic purposes, so how you are manage your insect pest in organic form?
How do I I could
pest,
not.
I am asking you that how you are manage your insect pest, especially
Oh,
the
right. Okay.
yeah.
Yes. That's a very, very, very good question. So first of all, we work with the organic farmers. So I don't do any research in the university premises because we wanted to adapt into what farmers do, right? So how they manage the diseases is very, very neat. So organic is like you don't spray anything. So you have to come up with the management system. So one thing is you have a cover crops, and then also all the bugs are going to be there like January and February. And then in the mid of the February, there's lots of ladybugs coming in and they eat all the insects. If it's, we don't have that. How they bring these ladybugs, they grow different types of plants in the patches around the field. These fields are large, but they have like marigold, they have some insect attract plants instead of spraying. Also, they keep their land very clean.
Okay.
Very clean means, yeah, they don't spray.
Okay. If you are saying this, that with their catch crops or with other attractive crops, they manage their insect pests.
Right, exactly.
Okay.
Crop rotation is the number one
So,
key is the crop rotation.
okay, that's nice. So, what about the chickpea pod borer? Heliopsis? Yeah.
Exactly. So this is a big issue for us, chickpea pod borer. So do you know how we can control that? Is the planting date. So if I plant in the third week of January, I have a pod borer in the first week of May. So what we did testing. We start testing from, can we plant in first week of December? So if I plant the first week of December, if I don't see a lot of temperature fluctuations, I could harvest them before the foot borer attack. So one of the thing, and then also we need to have resistance, and then probably we need to spray something like the USDA-certified. So pot borer is an issue. So growers are working on it, but daisies, Kaburis are the mostly affected by the foot borer. Daisies are not. So what we tried is the purple chickpeas, the daisy and the green chickpeas. They were not attacked by them. They were side by side, but they were safe. So we have to come up with ideas. We are working on it. And I also talking to pathologists and entomology people. We have a really big entomology department in our department, like the entomology group. So yeah, it is the foot borer issue.
Okay. One more question. That's on the end.
Sure.
Okay. Thank you. It was about the insect pest, and now I want to know about it. The weeds in pulses. How you are controlling, especially in chickpea.
Right. So the weed control is it's not a very big issue for us because our grower, like I said, we do the cover cropping and then we crimp it and we level it. Okay. So and then you need to talk to that expert of, I talked to Dr. Shirtlift from Saskatchewan. So it takes 5 to 6 years to reduce the weed seed composition in the soil. He came and gave a talk at our webinar series. So it's take 5 years. After 5, like you need to reduce the weed seeds in the soil. So our problem is for the pulses is the first 30 days until the canopy closure. So what we did was we ran cultivator 2 times. So we planted in a very clean soil. And then we ran the cultivator 2 times before canopy closure. Once the canopy is closed, really weed is not a problem.
Yeah. So it means, do you irrigate your field crop? Are you irrigate? Yeah. When you sow your
No,
chickpea.
we don't. We actually don't. Like, that's why we try to do this as a low input crop. We did not add any irrigation. We do have, if you see in my pictures of the field, we have irrigation system. If we want, we could irrigate, but I don't know why, because we have a lot of rain here. It's
Oh
humid.
yeah,
It's like,
okay. It's a myth. It's a myth. But
yeah.
in chickpea, if you have much more moisture, the genotype make more vegetative growth than the reproductive one. Is it? Do you face some such kind of problems there?
That's true. That's true. That's the material that I'm evaluating from Egypt is the most adapted one for South Carolina. And also from the Minico collection, we picked 30 different lines that actually they all are daisies, and the Billy Bean works very well. So those have, I did not see ascochyda and the big issues. So we have about like from we planted many times about 500 accessions, and then we shorten up for 30. So chickpea is hard, but I'm sure, because I have like a very experienced breeder working with me, Dr. Vandermark. So I think we'll be okay. We made quite a bit of progress in there.
So nice of you. Thank you so much. Thank you, Dev. Thank you, Nelson. Thank you.
Yeah, sure, thanks. There's a raised hand from Tom.
Okay, thank you, Dil. Very nice. I was curious that you talked about the FTMIR. Did Agilent develop the calibrations for you, or you developed them?
We developed it, and I actually hired a PhD program. So I have a PhD student. What happened was, so this project is part of the USAID. The breeders in Nepal, they wanted to measure protein content in the field. So that was an assignment given to me to sort this out problem. So NIR is good, but it's not sensitive to measure the other traits. So this FTMIR, it's a short unit, but it has a range quite specific for the traits that we're looking at. And then we develop all the models. So there's like 9 publications out from my program. If you Google my name, they will come. They all in the Plant Phenome Journal. All the models developed by us. But if you wanted to buy one, it's not that expensive, Tom. It's like $25,000. And then you upload into our cloud, you can get the data at the same time.
Okay, thank you.
So there's like 8 traits right now we can do, but we are trying to do for the flavors as well, because, you know, flavor is the hardest thing because they're very low concentrations. Yeah, but I would work with you on the model part. A couple of models. I think it has some sort of a trade, because once we developed the models, we had to give it to Clemson University. So they have some sort of a trademark in it. So you had to work with them, so they will give you the models.
Thank
So
you.
let me know. I'll give you.
Nice. We have also one question from the chat. So, have you tested breeding for adaptation to no-tillage condition in your growing environment? Is it considered part of organ breeding?
I couldn't get the question. Okay, how have you tested breeding for adaptation to no-till condition in your growing environment? Is this considered as a part of the breeding? I have not done for no-till because of the weed pressure, like Dr. Asid said. And all the growers here, we go for organic no-till system for sorghum. Because my rotation crop is sorghum. After I harvest the peas, they put sorghum in there or kale. No-till is very difficult for us without having weed control for the organic system. It is really nice. I don't know. It's too complicated with organic.
Okay. So, are there other questions? Nope. Doesn't seem. Okay, so I thank you again. Dil. So this will go on our website and YouTube channel soon, and see you all in I guess a couple of weeks, because we'll have. Let me just check it. We'll have the last webinar for our session this year on June 11. The dates were a bit changed. And with this, I thank you all, and see you. Bye.
Thank you.
Bye.
Bye, everybody.
Bye. Thank you so much. Thank you.